Good evening, everyone. Today, we will see in detail about the first step of glycolysis. So, in the last video, we saw about the overview, and today we will see the first step in little bit detail. So, what is the first step? The first step is nothing but glucose getting converted to glucose 6 phosphate. That means it is getting phosphorylated at the sixth position. And who's the enzyme responsible for that? We have hexokinase enzyme, and uh, it utilizes one ATP for the same. Right. So this is the first step of glycolysis, and this step has some significance. So the first thing to remember is that this is a priming the pump reaction. So this is the first step in preparatory phase where we are actually using ATP so that in the end we can gain some ATP out of it. So this is the first reaction of priming the pump. What is the second importance of this reaction is that this is a irreversible step. So it is irreversible under physiological conditions. So this is a irreversible step and it has a very high negative delta G. That's why it is an irreversible step. And the third thing to know about this is that this is a step in which glucose gets committed to metabolism. Committed to metabolism. So what do we mean by that? So what is the product here? The product is glucose 6 phosphate. Okay, only if glucose is converted to glucose 6 phosphate, then it gets committed to any sort of metabolism. Otherwise, what happens? Glucose enters the cell through a glucose transporter, and it is by a facilitated diffusion process. Now what will happen is that if the concentration is high enough inside the cell, it will start transporting the same glucose outside. So if you want to trap the glucose and some inside the cell, you have to phosphorylate it. So that is the first thing. So it is trapping the glucose molecules in the form of glucose 6 phosphate permanently inside the cell. That is the first thing. What is the second importance? The second importance is that it is this glucose 6 phosphate molecule. Now, once it is converted to glucose 6 phosphate, now it can go on and complete the glycolysis and produce pyruvate, and later pyruvate might be converted to acetyl CoA, and it can enter TCA cycle. So this is one way. Otherwise, this glucose 6 phosphate molecule can be used for glycogen synthesis, or it can be utilized for production of in the HMP pathway. It can enter the HMP pathway. So here you can either produce NADPH, or you can also produce ribose sugar. So pentose sugars can also be produced. In another pathway, it can be diverted to form triacyl glycerols, or it can also be utilized to form acetyl CoA and eventually synthesize fatty acids and cholesterol molecules. Or you can also convert this eventually into some sorts of amino acids. So all of these could happen only if glucose is converted to glucose 6 phosphate. So it is the first step which commits glucose to enter any of this metabolism. So that is why it is a committed step of metabolism. Then if you look into it, so this is a committed, that is why it is a committed step of metabolism. Now if you see, there is one another important aspect of this reaction, which is the magnesium. So let us see about hexokinase and its reaction. So now hexokinase is actually a kinase. That means it belongs to a class called transferases. 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 So they are transferring some group from donor to the substrate. So here, who is the donor? It is the ATP. And what is the group that is transferred? That is a phosphate group. And who is the substrate? Hexose. Hexose sugar. Hexose sugar or six carbon sugars. So it could be glucose, it could be fructose, it could also be mannose. Okay, glucose, fructose, mannose, it could be any of them. So it takes the terminal phosphate of the ATP and it is attaching it to the six carbon sugars. So that is the function of hexokinase. Now the donor is not just ATP. Instead, it is actually ATP magnesium complex. So if ATP is not complexed with magnesium, then this reaction does not happen. So ATP magnesium complex, magnesium is very important in this step. So what happens is that this ATP has lot of negative charges in it, and this magnesium somehow forms complex with them and diminishes these charges, so that it can go and transfer the phosphate group to a and form a bond with the hydroxyl group of OH group of 6 carbon of glucose. Now, this hexokinase, it is seen in almost all organisms, and it is also seen in all cells, and it exists as four isoenzymes in humans. So four isoenzymes. So what are isoenzymes? So they are isoenzymes. In the iso means same. So in the context of function, so they all catalyze same reaction, but they differ in their structure. So they are differ in their structure. So you can call them as different gene products that catalyze the same reaction. They are also encoded by different genes. So isoenzymes are different gene products that catalyze the same reaction. So in case of hexokinase, you have hexokinase 1, hexokinase 2, hexokinase 3, and hexokinase 4. So if they catalyze the same reaction, why do you need to have different isoenzymes? The reason is, even though they catalyze the same reaction, the rate at which they catalyze, the way they are regulated, these should differ according to the body's need. So that is why we have different isoenzymes. So of these four, the first three isoenzymes could be grouped together, and the last one, the fourth isoenzyme, could be grouped differently. So let us see about the first three isoenzymes. Their kinetics we will see first. So these are actually low Km enzymes. These enzymes have low Km. Now what is Km? Km is the substrate concentration at which enzyme achieves half of its maximum velocity, which is Vmax. So it is a substrate concentration at which enzyme achieves half Vmax. So here what happens is that it has a low Km. That means when substrate concentration is less, even then the enzyme could exhibit maximum activity. That is the meaning of low Km. And now if something has a high Km, that means for it to exhibit maximum activity, it needs larger substrate concentration. So we know in enzyme, first needs to come and combine to its substrate, only then it would catalyze that reaction. So enzymes which have low Km are considered to have very high affinity. So low km simply translates to high affinity. That means these have high affinity. That means even when the substrate concentration is very low, they are going to go and bind to it, and they are going to catalyze this reaction. So this first three groups of hexokinases, they are of high affinity type. But what is the problem? They are low capacity enzymes. So why are they low capacity? What do you mean by low capacity? That means their maximal activity is very limited. So the number of substrates they could catalyze per second is very low. Okay, so that is why they are considered as low capacity. So, on the other hand, we have this fourth isoenzyme, which is called as glucokinase. Now you can see the name itself mentions glucokinase. That means the substrate is only glucose. Okay, and the next thing about this is it has high Km, or in other words, it has very low affinity. So it has very low affinity. That means to activate it, you need very high blood glucose concentration. If you don't have very high glucose concentration, the glucokinase will not come and react. But even though it has very low affinity, this one has high capacity. So it has high capacity. What does it mean? So when the just imagine when the glucose concentration is very high and you have a low capacity enzyme, does it make any difference? Hexokinase was enough, because even at higher concentration hexokinase will still act. To activate it you need lower concentration, but even at higher concentration it will act. So if it acts at higher concentration, it has a low capacity. So why do you need another isoenzyme here? The reason why glucokinase is required is because it is a high capacity enzyme. So when the glucose concentration is very high, because of its high km, it gets activated. And once it is activated, it is a high capacity enzyme. So what it does is it could convert so many glucose molecules into glucose 6 phosphate molecules. Now, the first three isoenzymes, 1, 2, 3, they are seen in almost all the cells. But the glucokinase is only found in two cells. One is the liver, and the other is pancreas. Now, this is also very important. Now, what is a function in liver? See, liver has is a multifunctional organ, and according to the availability of glucose, it its function differs. So, for example, if you are in a fed state, okay, that means you have a lot of glucose. Your body has a lot of glucose. You have just had a meal and you are having a lot of glucose in your body. At this particular time, you have a lot of glucose, and liver functions actually as a glucose consumer. That means it consumes all the glucose because you have excess of glucose. So now liver has to utilize all these glucose primarily for oxidation, to get energy for its functioning, and secondly it has to utilize all the glucose for anabolic reactions, which we have mentioned in the last class. So glucose for production of glycogen, for the production of cholesterol, for the production of various other molecules, it needs energy. And for all and it also needs substrates from glycolysis and many other products. It utilizes all of these, and it acts as a glucose consumer. But in fasting state, it utilizes all the other non carbohydrate compounds and the glycogen that is stored to produce glucose for other cells. So, in fasting state, it becomes a glucose producer. So, it becomes a glucose producer. So, now we need to have enzymes that would cater for each of these needs. So now you see, when you have a lot of glucose, when you are going to utilize all the glucose, so soon after your food, what happens? All of the food is getting absorbed in your portal system, and they are first taken to the liver. So now liver has to try to extract as many glucose towards itself before letting all the glucose go to the other organs, so that it can use for all these anabolic processes. So that is where glucokinase comes into place. Now glucokinase, see, it has high Km. So in the after fed state, your blood glucose level is very high. So it would reach this Km levels. So it gets activated. So once it is activated, it is a high capacity enzyme. So it could, once it is activated, it will rapidly convert numerous glucose molecules into glucose 6 phosphate molecules, trap them inside the liver, and divert them to one of the anabolic processes. So in liver, the major function of glucokinase is to trap all the glucose that is required by the body for anabolic processes, maximum glucose, and then leave the rest to the other organs. But in fasting state, what happens? The glucose level in blood goes down. So the glucose that reaches the liver is very less. In that case, the glucokinase, the Km will not be achieved. So since it is a low affinity enzyme, it will not go and combine with the substrate only. Its activity will be very minor. So no need to worry in fasting state. Now on the other hand, what happens in the pancreas? In pancreas, it acts as the glucokinase we are talking about. It acts as glucose sensor. How? So when the glucose level in the blood increases, pancreas has to secrete insulin. So especially the beta cells. So it needs to have a mechanism to understand that the glucose is elevated in your blood sugar. Now if it has a normal hexokinase, what will happen? Hexokinase is a low capacity enzyme, so even when the level of glucose is very high, it would still only phosphorylate few of the glucoses. So the pancreas will not have an idea of how much increase of glucose is there in the blood sugar. So that is why it has glucokinase instead. So glucokinase, what happens when the glucose level increases, it becomes active, and it converts a lot of glucose into glucose 6 phosphate very rapidly. Once it does, all of this glucose 6 phosphate goes for oxidation, and it increases the ATP. The cellular ATP in beta cells is increased. Once the ATP levels are increased in the inside, what happens? It goes and inhibits or closes all the ATP sensitive potassium channel. Once it stops all the ATP sensitive potassium channels, that leads to depolarization of the cellular membrane and opening of calcium channels. So we'll write one by one. So if the blood glucose increases, all glucokinase becomes active, and it converts all of them to glucose 6 phosphate, and glucose 6 and eventually it causes increased ATP inside the cell. Now this increased ATP inhibits ATP sensitive potassium channels. This leads to depolarization of cell membrane, which leads to opening of calcium channels. So there is increased intracellular calcium. This leads to, whenever there is increased intracellular calcium, the one of the function of calcium is helping in the fusion of uh, granules, secretory granules with the cell membrane. So it helps in exocytosis. That process is called exocytosis. So it helps in that. So it activates hexocytosis. So what happens? The secretory granule membrane gets fused with cell membrane, and secretory granules is where all the insulin is stored. So what happens? It gets secreted outside the cell. So this is how in pancreas glucokinase helps. So we have seen different scenarios of function and the kinetics of each of these glucokinase and hexokinase. Now let us see little bit about their regulation. So all the hexokinases, especially 1, 2, and 3, okay, they have something called as end product allosteric inhibition. So what does it mean? Who is the end product of this reaction, hexokinase reaction? That is glucose 6 phosphate. When sufficient amount of glucose 6 phosphate is there, it will go and inhibit the hexokinase molecule, hexokinase enzyme. So it has end product inhibition. Now let us look into glucokinase. What happens to glucokinase? It does not have no end product inhibition. So what is the significance of this? Hexokinases have end products inhibition, but glucokinase doesn't have end product inhibition. Now imagine you have a cell that is very close to the liver circulation. So once all the portal system comes to the liver, then all the rest of the glucose that is not utilized by the liver will be given to the systemic circulation. Now the first few cells which receive this blood, if they do not have end product inhibition, what would happen? They will go on to phosphorylate all the glucose that comes inside them, whether or not it is required by the cell. And subsequently, the last few cells which are going to get the glucose will be left with very few glucose molecules. So to stop that, we have an end product inhibition. So once you have sufficient glucose 6 phosphate inside the cell, that means you have sufficient glucose 6 phosphate for all the metabolisms, all the sorts of metabolisms. Uh, so this is the starting product of almost all the metabolisms. So if you have sufficient, then you have to stop converting all the glucose so that the next cell starts getting enough glucose. So that's what happens here. Here you have glucose 6 phosphate itself, that is the end product, going and allosterically inhibiting the hexokinases. But if the same thing exists in liver, what would happen? It would inhibit or stop all the glucoses getting converted into glucose 6 phosphate for all the anabolic processes. So there you do not need this kind of checkpoints. So that is why glucokinase doesn't have an end product inhibition. So how is glucokinase regulated? So is there no regulation for it? Yes, it also has a regulation, and that regulation is with the help of glucokinase regulatory protein. Glucokinase regulatory protein. So uh, whenever uh, the glucose levels in the body or in the blood is high, okay, the glucokinase gets activated and it converts glucose to glucose 6 phosphate, and eventually glucose 6 phosphate gets converted to fructose 6 phosphate. Now when glucose levels are high, glucokinase okay, when the glucose levels are either when the glucose levels are low, this glucokinase is associated with the glucokinase regulatory protein, and it is present inside the nucleus of the cell. Okay, now when the glucose levels increases, this glucose level, increased glucose level, stimulates the release of glucokinase from the glucokinase regulatory protein. Okay, and then further down the glycolytic pathway, you have fructose 6 phosphate. When you have sufficient amount of fructose 6 phosphate, what does it do? It goes and stimulates the formation of this glucokinase and glucokinase regulatory protein complex. So what happens? It becomes inactive, and now it enters the nucleus and stays there. So now you can see a kind of regulation here. So whenever glucose levels are high, glucokinase, the increased glucose levels itself stimulate the release of glucokinase, and it remains active as long as this glucose 6 phosphate is being utilized for all the other anabolic processes. Once all the anabolic processes are taken care and liver starts utilizing this glucose 6 phosphate now for its own energy needs, what happens? We have increased fructose 6 phosphate. Now, now there is no need to keep on phosphorylating the glucose at that high level or high capacity. So what happens? The fructose 6 phosphate goes and it stimulates the formation of this complex where glucokinase is inactive. So this is how glucokinase is regulated as opposed to the hexokinase. So now here we can understand few things like one is, if you have a normal cell that is not a liver cell, increasing glucose transport need not necessarily commit all the glucose to for metabolism. So that is one important aspect that you need to understand. So simply by increasing the transport will not change anything, but that is not the same thing in liver, where increasing the transport might channel almost all the glucose to, or more glucose to, glucose 6 phosphate and subsequently to all the other metabolism because of these checkpoints. Okay, so this kinetics could be represented by a simple graph. So here we have enzyme activity, and here we have blood glucose concentration. Okay, so first let me draw for which enzyme? Hexokinase. So I'll take blue color. So this is how it will look like.
Hexokinase
Hexokinase
activity.
activity.
And this portion represents fasting blood sugar. Okay, this portion represents fasting blood sugar. So you can see in fasting blood sugar only, it has all achieved its Vmax. So you can see this is the maximum activity, and half of this is the maximum activity, and half of the maximum activity is this. So here is where the Km is. So the Km is much lower than the fasting blood sugar. So that is why it is very much active even in even when the blood sugar concentration is very low. Even then it is very much active. So cells can still utilize all the glucose that is present even though concentration is quite low. But on the other hand, let us draw for glucokinase. It will look somewhat like this. So here is where the Vmax for glucokinase. So here it is. So half would be here. So it comes somewhere here. So here is where the Km. So unless the blood sugar increases to this much level, This is the fasting range. So unless it increases to this much level, it does not even achieve the half Vmax state. But at very high concentration, it will achieve, and it is a high capacity enzyme. So this is how we can represent them. So now we have seen the first step, the first priming up step, and we know it is irreversible reaction. We know the mechanism of action. We have seen all the different isoenzymes and what are their functions in our body and how they are adapted to the function of the organs in which they are seen. I hope this was a very good lecture for you, that you have understood a little bit. So kindly comment in the comment section for your feedback, whatever it is. I will try to improve in the further session. Thank you very much.